In the introductions of our papers or in our talks, we justify plant phenomics for its role in breeding. However, it's probably worth spending some minutes to see to what extent this is correct and what is possible and perhaps what is not possible. For us, it's straightforward that phenotyping participates to breeding, but it's useful to mention that trait-based selection and phenomics is basically not in current breeding pipelines. This is a paper that explains that indirect selection has had limited success until now, and that the indirect method, and phenomics is an indirect method, is at best a complement to direct selection for yield. So in the breeders community, many people, perhaps the majority, thinks that phenomics is not terribly useful for breeding. And the question is, what can we answer to that? The key for that probably is that, okay, but what is the yield and the genetic control of yield? And this is probably key for that. So I present here the result of an experiment, a multi-site experiment in 16 fields, 2 years, 2 watering treatments over Europe. And we clustered all these fields in well water and cool, well water and hot, water deficit and hot, for instance. And you can see here that the yield was quite different depending on these scenarios, which was expected. The ranking of genotypes also differed between fields, so selection for yield then sometimes is more difficult. And if we do a genetic analysis of that, so here in the center you have a Manhattan plot with peaks. Each peak means that there is one gene or one portion of the genome that affects the yield. So we know that it affects the yield, but depending on the scenario that we defined before, the same gene can be highly positive in some scenarios, so here in hot day and night, and well negative in others, in severe water deficit and cool, for instance, here. So most QTL have positive, negative or no effect depending on scenario. So there is no positive or negative gene, it depends. And what is true for one QTL was basically true for the 20 QTLs of yield that we had all over Europe. So all of them were either positive or negative depending on conditions. If we take the last one, QTL 5, it's really positive in mild and really negative in water deficit. And if we take QTL 4, it's really positive in water deficit and really negative in the other one. So, yeah, most QTLs, most traits have positive, negative or no effect depending on the scenario. Whereas yield-based selection until now led breeder to select for traits that correlate with yield in on scenario. So this is the constitutive traits, which are good everywhere. So perhaps the idea is that the role of phenomics cannot be to predict or to improve yield, because breeders do better than us with a genomic prediction for yield. But perhaps they have focused on alleles that correlate with yield, whereas what we can do is to try to make use of gene of acclimation that were different, whose effect was different on yield in different scenarios, and so that do not correlate with yield regardless of conditions. So perhaps phenomics plus modeling could be a way to use those alleles whose effects are context dependent that breeder cannot easily use until now. So for that we have a major difficulty in that we know that the environment changes every minute basically, so the light can change by 1 order of magnitude over some minutes. It's the same for temperature, etc. So the mechanisms are defined for organs and over minutes, while yield is defined for a canopy of a month. So we have different scales, and here I defined three scales. So either minutes for cells or square centimeter, or minutes for organs, or month for canopies. And here on the left we can have the mechanism, short-term mechanism. In the center we can have mechanisms that are medium run, that both will be difficult to have in the field, but the field is the field, and here is the yield. So when we talk even of mechanisms, we don't talk exactly at the same thing. So we want to have the genetic variability of mechanisms at different scale, but what interests us at the end of the day is its effect on yield, but it depends on the upstream mechanisms. So I want to show some examples of what we can do with the phenomics indoor, either in indoor or in the field for mechanisms. So for instance, here we have phenotyping platforms for leaves and for roots, so we can build from images here some 3D plans on which we can perform calculations, for instance, of growth, but also, for instance, of intercepted light, intercepted water, etc. So how can we do that? We don't have these results from our platforms, but some calculation we can do that. So this is an example. With the image analysis we can have individual 3D reconstruction. And if I take this 3D plant from the field, but I put it virtually in, from the platform, sorry, but we put it virtually in a field, so then we can calculate a trait such as light interception. So this is something that will define the yield, that cannot be really done for hundreds and hundreds of genotypes in the field, but we can do that in this way. And if we can do that, we can do a genetic dissection and say that some genes are driving light interception, and then we can combine them with the other gene for other traits for calculating yield. And this is the idea. So this can be done for light interception, but it can be done for many other traits that I have no time to describe now, such as reduction use efficiency, even at some layers of the canopy, such as the layer where the ear is, for instance, in this example, by the rate of biomass accumulation, leaf area with a time resolution of minutes to day, depending on the platform, transpiration, stomatal control, components of water use efficiency. So for all of those, we can measure them indoor and calculate the traits that are originating, even traits that could be in the field by modeling, and then do the genetic analysis. Of course, we want to have some phenomics in the field as well. So it can be done by in different environments. Even we can manipulate the environment with a rainout shelter or with a phase. It's essential to measure this environment. And once we have done that, based on phenomobiles, drones, gantries, and all the imaging system possible, then we can calculate traits such as the plant height, the leaf area, etc. So we can have other traits difficult to have in the platform, in the field, and the other way around. So once we have both traits for indoor to field, do they connect at all? So is something that I measure in the platform valid for the field? And the response is, of course, it depends. But for some traits, platform can mimic the field, other it doesn't work. But for instance, you have the progression of leaf number in the field and in the platform. So you have 3 experiments in the platform and 2 in the field, and you can see that the same model accounts for both platform and field. So this is true for one genotype. it's the same, for instance, for response of water deficit. What I measure in a greenhouse or in a chamber can be valid. So this is the response curve. This is the air dryness, VPD. So 0 means it's wet, 3 that is dry, and the response curve is common to all these conditions, chamber, field, etc. So we can do indoor things that can be valid for the field. So there is hope, and if we can do that for one genotype, we can do that for many genotypes. So for here for 250, and this gives them the genetic variability of leaf appearance or the leaf the response of leaf expansion rate to soil water potential with high heritabilities. And if the heritability is good, this means that we can do a genetic dissection and use the genetic control of this gene to select the best possible alleles and combine these alleles for yield. So I would like to finish by showing how we can integrate all this information for actually selecting the best genotypes for each region. So this is an example in which we did genomic prediction of maize yield across Europe. And so first, all the panels, so there are 250 hybrids. We can have the progression of a plant cycle in the greenhouse, and I showed before that it's valid for the field as well. So we can know for each genotype the position of the vegetative flowering time and grain filling in time corrected for temperature. So then we can go to the field and apply that. So in each field the temperature is different, so the dates will be different, but we can calculate the position of each vegetative stage, flowering and grain filling for each field. And this will be different between genotypes, because I said that there is a genetic variability for that. So for each genotype in each field, we can position, for instance, here the flowering time. And if we can position the flowering time, we can calculate the light, the water potential, the temperature that every genotype sends in every field. If we do that, then we can build response curve of yield or grain number to light, water or temperature during flowering time or during other periods. And from that, these response curve were quite heritable as well, quite good. So we can write that the yield is a combination of all these conditions. But because our genotypes are genotypes, our hybrids are genotyped, so we have their genome, and from the genome we can calculate with the genomic prediction, we can train a model to calculate the slope. So each of these slopes are for real genotypes, but we can calculate them for new genotypes that are known only by the genome. And the genomic prediction predicts decently well the yield, so we can decide which is the genotype which is best fitted for North Europe, for South Europe, for Italy, etc. And it worked even for new genotypes and for new fields. So it's a way that is quite potent and usable in selection for predicting what a new genotype that is not phenotyped could give in Northern Europe, Southern Europe, etc. So we can combine alleles in something that is an ideal type. So my conclusion is that the role of phenomics needs to be rethought. It's probably not to help to improve yield everywhere, because again breeders do better than us for that with the genomics selection for yield. Probably our comparative advantage is to try to make use of adaptive alleles and traits that are almost not used in breeding until now. So in the field, it's difficult to disentangle the effect of environmental conditions. So the approach is first we measure yield in fields. So there is no idea of measuring yield in platforms, but we can measure the trait in platform in indoor or equipped fields. Then we can combine both yield and traits to train models of genomic prediction. And finally, we can predict the best combination of alleles for each scenario. So this combination of phenomics, genomic prediction, and modeling is probably key for using phenomics in breeding. And these are the acknowledgements, and I leave you with those.